With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello, let's solve this question. Which is not true about the PCR or the polymerase chain reaction? PCR can amplify very small amount of DNA. It can be used to detect HIV in suspects. Only RNA can be used as a primer, used to detect mutation in genes. So the first option is given. PCR can amplify a very small amount of DNA. Some of the steps of PCR are: first is the denaturation of DNA, of DNA. This is done by heating the DNA at about 42 degrees Celsius, where the strands of DNA are separated. Strands of DNA separated, separated. And in this denaturation process, after this denaturation, what is done? The second process is known as amplification. In this, a part of the DNA is replicated about 1000 times, 1000 times, and in this way the amplified gene is easily readable. Amplified gene is easily studied. So the first option, PCR can amplify a very small amount of DNA, this statement is correct here. Second option is given, it can be used to detect HIV in suspects. Option number B is also correct. Detection of HIV can be done by the analysis of blood. Blood analysis. So if PCR is done for the blood DNA, then it can easily detect the HIV in suspects. So this option is also correct. Option number 3 is given, only RNA can be used as a primer. This option here is wrong, because we know in the PCR method DNA primer is also used. DNA primer, which is known as the Taq polymerase or the Thermus aquaticus Taq polymerase. And it sustains in very high temperature conditions, and this DNA primer is used. So this option, only RNA can be used as a primer. Both DNA and RNA can be used as a primer. The last option is given, used to detect mutations in genes. Obviously this PCR is used to detect mutations, and these mutations can easily be studied if a gene which is mutated is amplified 1000 times. So this option is also correct. So we have to find out not true about PCR. So option number 3 will be the correct option here. Thank you very much.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.